All right, today we're going to talk about transcription, translation, and a little bit on mutation. I don't think we're going to get to it all today, all right, because it's a long lecture. There's a lot to know about transcription and translation. And on that note, all right, I have made the executive decision to not talk about epigenetics. All right, so that was what we were going to talk about on Friday. We're not going to talk about that. I don't expect you to know it, so I know it's on the study guide. Don't even bother with the questions because we're not going to go over it. All right, there's enough content as there is. yes. Now, I might do a couple slides on Friday just telling you what it is, but we're really not going to go in any depth. I might spend 5 minutes on it. All right, so don't worry about epigenetics. Instead, on Friday, we're going to finish up this lecture. We are going to do some worksheets, and there might be a potential to earn some exam points. All right, so I would not miss Friday. All right, any questions before we continue on? All right, this is another long, dry subject. I try to break it up a little bit with some questions, so if you have any questions as we go, just let me know. All right, so we're looking at transcription and translation. All right, how do we get from DNA to an actual working protein? All right, so we can look at gene function at basically these two different levels, the molecular function and the organism's trait. All right, and they work together. The molecular function affects the structure and the function of the cells to determine the trait that we are seeing. All right, and that brings us to the central dogma. All right, so you've probably heard this before, the central dogma. When you hear this term, we are referring to the process of transcription and translation. All right, so transcription, we are looking at a piece of DNA, and we are transcribing that into an RNA copy, specifically mRNA, or messenger RNA. All right, so this messenger RNA is then going to go on to go through translation, right, where we're taking the nucleotide sequence, an mRNA, and translating it into amino acids and ultimately a polypeptide and a protein. All right, so we're going from DNA to RNA to protein. That is our central dogma. Right, now there's some differences between prokaryotic and eukaryotic cells in how they use the central dogma, or how they carry it out, I should say. All right, so if we look at a prokaryotic cell, we have our bacteria cell here, we see transcription and translation both happen in the cytoplasm. All right, they're both happening in that cytoplasm, where in eukaryotes, we have half of it happening in the nucleus, the other half happening in the cytosol. All right, so in bacteria cells, transcription, translation, cytoplasm. Eukaryotes, we have transcription, RNA modification happening in the nucleus, and then outside of the nucleus, we have translation. All right, so these eukaryotic cells, we have that additional step of this RNA processing. All right, so essentially we're taking DNA, turning it into pre-mRNA, turning that pre-mRNA into RNA, right, and then translating that into a protein. All right, so there's that extra little step there. All right, so let's start with transcription. So when we're looking at transcription, we're really looking at these genes, right? We know that's an organized unit of nucleotide sequences that enable a segment of DNA to be transcribed into RNA and ultimately that final product. All right, so we are taking a look at the gene on DNA, we're making it into RNA to eventually be realized as the final product. Now, typically we have the final product being proteins, all right, but sometimes that final product is going to be the RNA itself. All right, so we can have the mRNA making a protein. We can have the mRNA simply being the transfer RNA or the ribosomal RNA. All right, transfer RNA translates the mRNA into amino acids. We'll talk about this. And then ribosomal RNA is actually a catalyst for forming polypeptide bonds, and it's part of ribosomes. I'm going to keep moving on because these slides are at your convenience here. All right, so let's look at transcription in a bacterial genome. All right, and we're going to start and mostly talk about bacterial genomes because they're less complex, but the basics are the same between bacteria and eukaryotes. All right, so if we're looking at a bacterial genome, there's a couple different segments within the DNA that are recognized. All right, so one is this promoter segment. Right? It's a sequence of nucleotide bases that signals the beginning of transcription. All right, so it is telling our organism to start transcription here. All right, at the other end we have the terminator sequence. All right, it's telling our organism to stop transcription here. All right, and everything in between the promoter and the terminator is going to be that transcribed region. Right, that's the region we're turning into messenger RNA. Now we also have this region of regulatory nucleotides, regulatory nucleotides that will kind of regulate how fast we are transcribing our mRNA. All right, so it can upregulate this, make it transcribe faster, or deregulate it. All right, that's all we're going to go into for that. All right, if we talked more about epigenetics, we would go a little bit further into regulatory sequences. All right, so if we look at transcription, there are 3 different stages: initiation, elongation, termination. All right, it's the IET. You can make your own acronym out of that if you would like. IET. All right, so the first step is initiation. And this is the recognition step. All right, so in bacteria, we have this sigma factor. All right, it's this protein here. This sigma factor is going to bind to the RNA polymerase. All right, and that's going to cause this RNA polymerase to recognize the promoter region. Remember, that promoter region is where we start transcription. All right, now this stage is completed when the RNA polymerase has bound to the promoter region and we have this open complex. All right, we've separated 2 strands of DNA. All right, in the next step of elongation, right, we have RNA polymerase actually synthesizing the RNA. All right, so we've separated out the 2 strands of DNA here. Now one strand is going to be used, and that's going to be called the template strand. Right, that is the strand we are using to synthesize our RNA. Right, then we have the other strand of DNA that we call the non-coding strand, right, because we're not coding anything off of that one. Now if we look at synthesis here, all right, we're going to do synthesis in the 5' to 3' direction. Right, very similar when we looked at DNA replication. The only difference here is we're making mRNA, all right, and we're using uracil instead of thymine. All right, so we've made our RNA at this point, and now it's time to terminate transcription. All right, so this RNA polymerase is going to reach this termination sequence here, telling it to stop transcribing. It's going to cause the polymerase to detach from the DNA, and it's going to cause this completed mRNA transcript to detach away from the DNA and the RNA polymerase. All right, we have our completed mRNA. All right, so this is basically the same process in eukaryotes, but eukaryotes, it's a little bit more complex and contains many more proteins, all right, which I don't expect you to know, but I do expect you to know that it's basically the same, a little bit more complex. All right, so just to kind of recap and make sure we're getting this all into our brain here, answer these couple questions as a group, and then we'll come back together.
Initiation, the Attached to them. Is it synthase? Is it not?
It ends in ace.
Polymerase.
Yeah, it's
What
going on
polymerase?
that.
RNA.
Also, Mutation is told me that
Oh.
my dad ordered it at the same time.
Same time?
So, like, the first time I had it, huh? Talking about how we're leaving at the same time. Oh.
It's a long drive. Where is that game?
Indiana, Iowa, it's like a 4-hour drive.
Dang, must be like southern part.
It's like just south of Des Moines. Yeah. And last time we were on the AC broke on Saturday.
Oh.
But it was like the worst. 75 degrees.
Yeah.
And we like did like the temperature thing. It was like east 85 on the bus. Driving back for like 3 hours, 2 hours drive.
Oh, maybe that might work in your benefit this time.
Right. There is that. True.
I like this. This is a lot simpler of a thing.
Yes, it is.
That makes me happy.
All right, so let's go through this. All right, what is the first stage in transcription? Initiation. And what happens in initiation?
No, RNA polymerase attaches the DNA.
Yeah, RNA polymerase is going to attach to the DNA at what site in the DNA? The promoter sequence, yes. All right, so we have attachment at this point. We're initiating transcription. The next step is making it elongation. Yes, so what is happening in elongation?
Getting elongated.
Yes, the RNA is getting elongated, right? Transcription is actually happening. We are making mRNA, all right? And we're continuously elongating that because we're adding on those nucleotide sequences, all right? Instead of thymine, what base are we using? Uracil, yes. All right, and the last step, which is termination. And what happens in termination? Yeah, everything lets go. The polymerase, RNA polymerase detaches. The transcribed mRNA detaches away and is now completed. All right? And we kind of gave it away. What is the name of the protein used in transcription? Polymerase, but what polymerase? RNA polymerase, yes. All right. So I mentioned that between eukaryotes and prokaryotes, eukaryotes have this other step, all right, where we have to process the pre-mRNA, which was just made in transcription, into the actual mRNA. All right, so in bacterial cells, this can happen immediately. We can go from transcription to translation. Eukaryotes, we have this intermediate step. All right, so we have to process it into the mature mRNA. So if we look at the pre-mRNA, we have these two alternating regions, essentially. We have introns, which have been transcribed, right, but they are not going to get translated. And we have exons, right? Exons are the coding sequence that we are actually going to find in the mature mRNA, and is the coding sequence that codes for genes. All right, so a good way to remember this, it's the intervening regions, introns, all right? Or the exons is the expressed regions, or exons, all right? So in order to do this, in order to get rid of those introns, we have to splice, all right? So we're going to go through splicing, get rid of the introns that are not coding for those genes. Now there's also going to be other modifications, such as adding on tails and caps to our mRNA. But let's look at splicing first. All right, so here we have our DNA, all right, with our promoter region, our terminating region, right, and we have these exons and introns. All right, now they're initially all going to be transcribed into our pre-mRNA. Right, but now the eukaryotes are going to go in and splice this pre-mRNA to make mature mRNA. All right, and the mature mRNA only contains the exons. All right, now in the meantime, we're also capping and tailing our mRNA to make it mature. All right, so what does this mean? All right, so capping is where we are adding a modified guanide attached to the 5' end. All right, so this cap is needed for the mRNA to physically exit the nucleus, and it's also needed for the ribosome to recognize it and to bind to the ribosome in the process of translation. All right, so this is helping us pass through the nucleus and being recognized by the ribosome. Now we're also adding on a poly-A tail. All right, so a poly-A tail is a bunch of adenine nucleotides, 100 to 200 of them, added to the 3' end. All right, and the purpose of this is just to increase stability and lifespan in the cytosol. So as soon as this exits the nucleus into the cytosol, it's not going to immediately break apart. This poly-A tail helps it stay together a little bit longer. Right, now this is not going to be encoded into a gene. Right, this does not code for any gene. All right, let's come back together and synthesize what we just learned and answer these questions.
The tail is because like it gets caught in things and stuff, so if it gets caught, it's not like interfering with important information.
Genetics is not my strong suit. I know very basic genetics. So that could be. It would make sense, yeah. No, you had it right. Yeah, so transcription happens first, and we're transcribing the DNA to RNA. Now we're translating the RNA to an animal. Like DNA, like be an enzyme. Other processes down the road. Transcribe for RNA polymerase. Okay. Yeah. Cool. Yep. Alright, so this should be a fairly easy one to answer. So let's come back together here. Alright, who wants to briefly describe the process of splicing? What does splicing do? We're removing something. What are we removing? Introns, yeah. So we're removing the introns from the pre-mRNA. What about when we add a cap? What is that cap?
So it can travel out of the nucleus.
It's a modified guanine, yep. And so it can help it travel, yep. And it also helps it, make sure I'm thinking about this right, because I just mixed myself out. Yes, helps it exit the nucleus, but also bind to those ribosomes. I want to make sure I have that correct in my head. All right, and what about a tail?
Junk on the end.
Junk on the end, what is that junk? Adenine, yeah, it's a poly-A tail. It's a sequence of these adenines, and what is the purpose of that? Increased stability, yes. All right. Now, does RNA processing occur in prokaryotes? No, I should have heard a resounding no there. Yes, this does not happen in prokaryotes. It's only in eukaryotes. All right, any questions? All right, let's continue on. All right, so once we're done with transcription, we can start talking about translation. All right, and this is where we start talking about the genetic code. All right, so the genetic code is a sequence of bases in an mRNA molecule, and they're going to occur in groups of 3. All right, so they're going to occur as a triplet called a codon. Right? So if you've listened to the podcast for the next assignment, you might have heard them talk about these codons and these triplets. Right? There's an interesting experiment that they did to try to figure out which codon codes for a particular amino acid. Right? And that's one of the questions on your assignment. So I'm not going to cover it now, but know that each group of 3 nucleotides, a codon, specifies a specific amino acid. Now they're also going to specify start and a few stop codons. All right, so there's some commonality among these codons. All right, they are all degenerate, meaning that more than one codon can specify the same amino acid. All right, so if we look at the very top here, we see that the codon uracil, 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 or UUU, all right, codes for phenylalanine. All right, but so does UUC, also codes for phenylalanine. Right, so more than one codon can be specified for an amino acid. It's also unambiguous. Right, so a single codon is never going to code for more than one amino acid. Right, so UUU is always going to be phenylalanine. All right, CUU is always going to be leucine. All right, so leucine can have multiple codons, but one codon is only going to have one amino acid. Right, they're also non-overlapping. Right, so they're very distinct. CUU codes for leucine. We don't start at the second letter for the next code. All right, we go on to the next letter. So I think, let me write this out because this might make more sense. All right, let's do CUU for leucine. All right, we're not starting and looking at UU, whatever's next. We're not starting at the last one and going U next. All right, we're starting all over. There is no overlap here. So let's say the next one here is valine. GUU. All right, so there's no overlap. They are very distinct. Finally, they're conservative. So if we look at each amino acid, all right, those first 2 nucleotide bases are going to be the same. All right, so let's look at leucine. They all start with a C and a U. Right, and this is helpful that if we get mutations that happens in that 3rd position, right, it's likely that mutation can still code for leucine. Right, there's a higher probability that it's not going to cause a significant change in what it's coding for. Right, so all these codons kind of abide by these rules. All right, so let's look at translation. And we're going to look at translation of bacterial mRNA again. All right, so the first thing that we have is this ribosomal binding site. All right, so this is the site that the ribosome is going to bind onto the mRNA in order to start translation. All right, now this is what bacterial cells have, is this ribosomal binding site. All right, but eukaryotes have that guanosine cap, that modified cap. All right, so that is what the ribosome will recognize for eukaryotes. Now it's going to find the start codon, which is typically AUG. All right, that's going to signal that this is the start of where you should translate. It also has a stop codon, signaling where we should stop translating. All right, and everything in between is that coding sequence. All right, so groups of 3 are going to code for different amino acids. A series of codons from the start codon to the stop codon. That is our coding sequence. Right? This is what's encoding for a protein. Right? Sometimes those stop codons could be called termination or nonsense codons. All right, so they kind of have more or less a numbering system. All right, so the start codon is going to define where we start. All right, so if we look at this sequence here, if I was a ribosome, I'm not going to detect that I'm starting to translate anything until I get to AUG. All right, AUG is going to tell me to start. All right, then I'm going to go in groups of 3, all right, and they're each going to line up to their specific amino acid. All right, now sometimes we can get mutations that insert in a nucleic base, all right, a nucleotide base here. So in our example on the bottom here, a U has been inserted. All right, now because we're reading in groups of 3 and we've inserted an extra base, right, our code completely changes in what amino acids it's going to encode for, right? So our amino acid sequence down here is much different than our amino acid sequence that it should be, right? And this is a type of mutation called a frame shift mutation. All right, some more questions to break up the monotony here. All right, answer these 3 questions as a class and we'll come back together.
Hold on. Oh wait, start is always a stop. Which one is the
Only 8. Yeah, RNA processing. That's an RNA processing, and this is in trans.
They all end in C, right? Like val.
For the most part, yeah.
Would be val.
It could be like thymine, but that's valine.
Oh,
So they all end in I and E.
valine. Okay. Okay, I have a question.
Yeah.
What's the difference between start codon, tail, cap, and promoter? Are those all the same thing? Because they all have to start, right?
No. So promoter says start for transcription. Okay. Right. So promoter's on codon on arm. Starting translate.
Okay, hold on. RNA, start translation.
RNA, the start codon is for translation.
Okay. And then promoter is DNA. That's first translation.
So the cap, that is the start here. The cap is with the ribosome group, right? We'll look at that a little bit.
Is the tail the first part? Okay, so then what?
It's like a stop codon before the tail. Tail's not going to do anything. It's just there to help keep your ability to move. All right, so what do we call a group of 3 bases that code for amino acid? Codon, yes, it is right up on the screen. All right, so what is the start codon? AUG, all right, so that should be the magical codon. If I gave you a string of nucleotide bases and asked where would I start translation, right, you should be able to pick out AUG, all right, it's the only start codon. All right, now what are the possible stop codons? Yeah, so there's 3 possible stop codons, all right? So if we look at a codon that is GUA, what amino acid is it coding for? Valine, yeah. It's down on this bottom here. GUA codes for valine, all right? Val is the abbreviation for that. All right. So beyond mRNA, which we have our codons, right? And we're using a U instead of T for our nucleic bases, we also have tRNA that plays a huge role in translation. So tRNA has an anticodon, right? So this is 3 RNA nucleotides, all right, that are going to be part of the tRNA molecule. And this is what allows for binding of the tRNA to mRNA, which is an important step in translation. Right, so if we look at transcription and translation, right, we have our DNA up on top. All right, from our DNA, from our template strand here, we are getting our mRNA. All right, once we have our mRNA, we're going to use those codons to find the matching tRNA. All right, so if we look at this codon in the middle here, UUC, all right, we are finding a tRNA molecule, all right, that has an anticodon, all right, it's complementary to the codon of the RNA of AAG, right? So this is how we're pairing tRNAs to the mRNA. Now this is important because it's the tRNA molecules that carry these amino acids so that they can be linked together to form a polypeptide. All right, so if we take a closer look at what tRNA looks like, all right, it typically has some common features in that we have this kind of clover shape to it. All right, there's going to be a part where that has the anticodon, which is just 3 nucleotide bases, RNA bases. All right, notice that we can have U in our anticodon, so we know it's RNA. And then we have this part up at the top, that's the acceptor stem for the amino acid binding. It's at the 3' end. This is the part of the tRNA that's going to bind to an amino acid so that it can bring it to the ribosome and create a protein. All right? So for example, if we had tRNA with the superscript SER, right, that is a tRNA molecule that has the amino acid serine at that 3' end. All right, so how do we get those amino acids onto our tRNA molecules? All right, so we're using an enzyme called aminoacetyl tRNA synthetase. All right, we're catalyzing the attachment of the amino acid to the tRNA molecule. All right, and in this step, I don't expect you to know exactly how it happens, but we are using ATP, all right, to bind the amino acid to this enzyme. The tRNA molecule comes in, all right, binds to that amino acid, and the result is a tRNA that we would now refer to as a charged tRNA or an aminoacetyl tRNA. All right, so if you see charged RNA or an aminoacetyl tRNA, we know that it has an amino acid attached to it at this point. All right, let's also pause again here and talk about tRNA.
What's the point of the
It's so we can recognize the codon. So when we actually get to the process of translation, basically we have a codon.
Okay.
Codon mRNA is going to be complementary to tRNA. Yep. Yeah, they're complementary.
Yeah.
All right, we're getting quiet here. I'm assuming that means we know exactly what a tRNA molecule looks like. All right, so what is the general structure of this molecule? Clover shape, yeah. And what might we find on this clover shape? An anticodon, yeah. So what is an anticodon? Opposite of codon, yeah, essentially. All right, we have 3 bases of mRNA together. That's going to be complementary to a codon on the mRNA. All right, so that is the anticodon. All right, at the other end of this clover shape, we also have another structure. What would that be? Yeah, the attachment site for the amino acid on that 3' end. All right, so that's where the amino acid is actually going to bind to the tRNA to make it a charged tRNA. All right, so what is the importance of having an aminoacetyl tRNA synthetase? What does it do? What does that enzyme do? It picks up an amino acid, and where does it put it? On the tRNA, yeah. So it's adding that correct amino acid to the correct tRNA molecule to create our charged tRNA molecule. All right, saying tRNA too many times today. All right, so what would happen to the cell if that enzyme became defective? What might be the result? What can't we do? We can't translate because we don't have the amino acids on the tRNA molecule. All right, we can't add it to the tRNA molecule. And this was a kind of a reaching question that's going to lead into what we're looking at next, because that tRNA molecule plays a critical role in translating that gene into an actual functioning protein. All right. So the last major step we're going to look at is the actual process of translation. All right, and we have to use ribosomes. All right, so here again we have another difference between prokaryotes and eukaryotes. Prokaryotes, they have one type of ribosome. Right? Eukaryotes, they have distinct ribosomes in different cellular compartments, most notably in their mitochondria and in their chloroplasts. They have different ribosomes. All right. Now, ribosomes, they compose of a large subunit and a small subunit. And between, say, prokaryotes and eukaryotes, so bacterial cells and animal cells, let's say, all right, there are some structural differences. All right, and we can use these structural differences to be exploited by antibiotics. All right, so antibiotics are going to be able to bind specifically to the ribosomes of bacteria instead of to bacteria and animal cells, because bacteria has that different structure to it. All right, so we're exploiting this difference in structure when we are using antibiotics to inhibit bacterial ribosomes only. All right, so I mentioned the structure of the ribosome. We have a large subunit and a small subunit. All right, but we also have these distinct sites within the ribosome. All right, so we have our P site, we have our aminoacetyl site, and we have our E site, or the exit site. And we're going to see how this works with the tRNA molecules to create a protein. So just like transcription, we also have 3 stages for translation, and they happen to be called the exact same thing: initiation, elongation, termination. All right, so let's look at initiation for translation now. All right, so this is where we have the mRNA is going to bind to the ribosomal subunit, the small one here. And that's going to be facilitated by this ribosomal binding sequence. All right, so it's this binding sequence that'll help it bind to the ribosome. Likewise, if we're talking about eukaryotes, all right, we know that it's the cap, that modified guanine cap, that will allow it to bind to the ribosome. All right, so once it binds, that start codon is not very far away. It's just a little bit downstream. All right, an initiator RNA, tRNA molecule, is going to recognize this start codon, and once it recognizes it, the large subunit is going to attach, all right, and we can start on with translation. All right, so at the very end of this step, we have our subunits attached, we have an initiator tRNA molecule in the P site, and we are ready to start translation. All right, so translation, we are using these charged tRNA molecules to add on our amino acids. All right, so the aminoacetyl tRNA is going to come into this A site, all right, and bring a new amino acid that is coded for on this mRNA. Right? So this mRNA is coding for this anticodon, which is bringing this specific amino acid to it. Right? So it's going to enter in this A site. A polypeptide bond is going to form, right, between the amino acid and the A site and that growing polypeptide chain that we see in the P site at this point. All right, so this isn't the first tRNA that comes in. We're a little bit downstream from where translation started. We already have some amino acids formed together into a polypeptide. We're transferring that polypeptide onto the new amino acid, right? Making it longer, elongating it here. The last thing that's going to happen is this ribosome is going to shift over, right, and shift the positions of these tRNA molecules so that the one that already gave away its amino acid can exit. All right, it's in the E site. It can exit away now. The one that we just bound the new amino acid to is going to move into the P site, right, and that's going to leave this A site open for the next amino acid to come in and bind to the next codon on our mRNA. All right, so this is how we're elongating, making this polypeptide. When we get to a stop codon, we're going to terminate. All right, so the last step, termination. We are recognizing one of 3 stop codons. It's going to be recognized by this release factor. Right, so when we get to a codon, this release factor comes in. It tells the polypeptide to be released from the tRNA. tRNA is going to leave, and the ribosome is going to break apart. Right, so that's the end. We have our completed protein, our completed polypeptide. All right, now I work better with visual cues for this. So we have a short animation that shows this, just kind of putting it all together.
The second step of translation is called elongation because our peptide is going to start getting longer. After the initiator transfer RNA attaches to the P site, the second transfer RNA, which is carrying another amino acid, settles into the A site on the ribosome. If the second codon is GCU, for example, the second transfer RNA will have an anticodon of CGA. This particular transfer RNA would be carrying the amino acid alanine. An enzyme in the large subunit of the ribosome transfers the methionine from the initiator transfer RNA to the alanine bound to the second transfer RNA. A peptide bond forms between these 2 amino acids, forming a dipeptide. Once this happens, the ribosome slides on to the next codon on the messenger RNA. This action shifts the initiator transfer RNA to the E site, and the second transfer RNA to the P site. Having given its amino acid to the dipeptide, the initiator transfer RNA will exit the ribosome and seek out another methionine in the cytosol. The next transfer RNA will settle into the A site. Once again, the enzyme transfers the amino acid at the P site to the transfer RNA at the A site. However, this time, both amino acids of the dipeptide transfer over, making the peptide an even longer chain of amino acids. This process continues until the ribosome reaches the stop codon, at which point a protein called release factor binds to the A site, rather than transfer RNA. The new protein releases from the ribosome, and the ribosome disassembles. This last step is called termination, and our new protein is now ready for packaging and export.
All right, so hopefully that visualization kind of put it all together there. And I think a good way to remember these sites here, because they're each labeled with a letter, EPA, Environmental Protection Agency. All right, that's how I remember it. All right, it's a good acronym here. And one thing I should mention is sometimes RNA codes for other RNA, right, which we saw for tRNA. It also codes for rRNA. And rRNA is used in this process specifically in elongation, to catalyze this new peptide bond, right? So that's where we are using rRNA.
Is the order of the sites, is there like a specific order?
It always goes EPA.
Okay.
Yes. All right. Let me just check the time. We have a couple minutes here. All right. So if we're looking at our sequences here, differences in our codons can relate to differences in what we see in our phenotype. All right, so if we're looking at our actual DNA, all right, we're transcribing, translating, getting our proteins into a polypeptide. In this instance, we're looking at a mouse with a dark coat. Right, now if we have just one difference, right, that's going to lead to a whole different amino acid being bound. And we see here that that leads to a different phenotype, right? We have a phenotype of a mouse with a light coat, right? So small differences can change our phenotype. Now, if we look at our sequence of RNA nucleotide bases here, we can get some mutations that occur. All right, so we can get a silent mutation. So if we have a mutation here, that's not what we would expect it to be, not what the original was, right? But it is different. It might code for the same amino acid, right? Remember, the first two bases are always going to be the same. That third one changed. It can code for the same amino acid. We might get a missense. All right, so that means we have this base substitution that changes one amino acid, changes the resulting, or changes one base, changes the resulting amino acid. Nonsense can shorten the polypeptide. All right, it's telling the ribosome to stop, essentially. And then we can have a frame shift mutation, which we talked about a little earlier. We're inserting an extra base instead of substituting one out, and that's shifting how we read the rest of the mRNA. All right, so each of these types of mutations can lead to different polypeptides. All right, so I'm going to end class here, and we will come back to the last two slides on Friday. And Friday we will have a lot of in-class work to help us understand and get ready for the exam. Could be. There's a chance for exam points. You never know what that might be. Tossing around a couple different ideas.